Hello, everybody. This is Dietmar Schwartz with our first online lecture. So I'm going to make it full screen. So welcome to genetics, everybody. I'd rather teach this in person in front of the classroom, but I actually don't think this is going to be too bad, given that genetics will involve a lot of independent work and problem solving and things like that. So this may actually work out rather well. So genetics is really at the very center of biology. And biology is the science of studying life. And as biologists, we should try to define life. So one definition, and there are many, is, for instance, life is composed of organized matter that is capable of undergoing reproduction and natural selection. And actually, I highlighted reproduction and natural selection. Actually, the organization of the matter is also impacted by genetics. So all of these, basically all aspects of life, the organization of matter, right? How the body basically turns genetics information into an organized body, reproduction, of course, and also natural selection, right? We need to encode the variation. So life doesn't really work without genetics, and therefore this class is basically central to all of biology. And humans have asked genetics questions for a long, long time. And the basic question is, right, why do offspring look like their parents? And this is like a really fundamental property of life that we don't really think about, but how does it come that chicken zygotes develop into other chickens and not puppy dogs, as in this case, right? So how are organisms breeding true to type, to use sort of a 19th century explanation for this? And this also relates to individual variation, right? So we not only know that humans will have will have human babies that will grow into humans that also have human babies, but we know that offspring tend to look like their parents. And here is a nice set of pictures that shows like either siblings or parents and their offspring. And so how is this variation that we see among organisms actually passed on to the offspring of these organisms? And humans have known and manipulated about this genetic variation for a long time. So what you see here are all different kinds of cabbage. So this humble plant up in the back is something that you would find growing in northwestern Europe on the beach. And this is actually cabbage. All of these things belong to the species Brassica oleracea. And through successive breeding and selection, this this plant has been turned into all of these varieties that you hear and that you may love or hate to eat. And genetics is really also the key puzzle in explaining evolution, right? So if evolution is descent with modification, both the descent part, right, the reproduction, the faithful reproduction of one parent into their offspring, as well as the variation that comes in with modifications that we know today as mutations, are all based on genetic mechanisms. And it's sort of an irony of history that Mendel's ideas were already known or should have been known at around the time that Darwin was publishing his work, but were basically ignored by scientists at the time. And biology may actually be ahead by several decades had that connection been made. And also, genetics is really key to 21st century biology. And this may actually have profound, and probably with profound societal consequences. And there's really like two main parts to this. So we have both genomics, so our ability to look at genetic variation at the genome level, has resulted in really powerful techniques to start looking at the genetics of biological traits. And then on the other hand, we've got biotechnology, not a major focus of this class, but related, that allows humans to change the genetic information of organisms and to make use of this information on biological variation. And I'd just like to point out two examples of how this is actually starting to affect humans. So the first example is pre-implantation genetic testing. So you may have parents that know that they're actually carriers for a genetic disease. So one of the famous cases is Huntington's disease, which is a dominant disease. So somebody who's heterozygous will develop the disease if he has the right sort of disease allele sometime during their 40s, 50s. And it's a fatal disease. But nevertheless, some of these people want to have children, but don't want to pass on that harmful trait to their offspring. And there is a way to make this happen. And this is basically combining genetic testing with in vitro fertilization. So in the first step, we have an egg in the case of humans that has 23 chromosomes, and the sperm comes in also with 23 chromosomes. So we have a fertilized egg, a zygote with 46 chromosomes. And now cell divisions start within the embryo. And at first, as you may know, the divisions don't add, the cells don't grow, the cells become smaller and smaller. And at a certain stage, there are enough cells present that scientists can actually remove one of these cells. And at this early developmental stage, the removal is actually not harmful to the development of the embryo. And what can be done is that this cell here now carries the genotype, the genetic information of the embryo, and can be used for testing, whereas we can sort of keep the embryo and let that embryo divide. And after the genetic test, we will know which embryos actually have inherited the harmful allele and which embryos will not have inherited the harmful allele. So if, let's say, mom was heterozygous for the Huntington's allele, half of her eggs should have carried the Huntington disease allele and half should have carried the wild type allele. And so on average, we would expect a 50-50 chance of picking an embryo that does not carry the Huntington's disease allele. And these embryos that don't carry the harmful allele can then be used for in vitro fertilization. The second example is, of course, famous CRISPR-Cas9. And in this case, I'll just encourage you to watch this short video. And on Monday, we will actually discuss this in class, in our online class, when we meet again in person, so to speak, on Zoom. So I'm going to try to use a learning progression in this class. And the first big idea that this lecture will address is the following: both genetic and environmental variation shape phenotypic variation, right? So it's both the genetics and the environment that shape the phenotype. And our learning objective is that for complex traits, phenotype cannot be predicted from genotype, as phenotypes of complex traits are the result of variation at multiple genes, environmental variation, and the interaction between genetic and environmental variation. So I'd like to introduce this with an example that involves voles, these cuddly animals here, and humans. And in this case, this is an example from behavioral genetics. And there's two closely related species of voles. On the one hand, we have the prairie vole, who lives in sort of the Midwest of the US, and the meadow vole, who lives on the East Coast in sort of forested areas. And they're very closely related, have very similar lifestyles, but there's an important behavioral difference when it comes to pair bonding. The prairie vole is socially monogamous, what that means is the male sticks around and actually helps the female in rearing the offspring. This is only social monogamy because the male may still undergo matings with other females, and the female may also mate with additional males. Um, but when it comes to the social interactions to pair bonding, they are basically stay together as a pair. Whereas in the meadow vole, it is basically the female that takes all the work of rearing the offspring, whereas the male basically both has multiple matings and does not engage in pair bonding and care for the offspring. And we refer to that as socially promiscuous. And in meadow voles, right, we can raise these animals in the lab, we can do genetic manipulations with them, is that the difference in the pair bonding behavior is linked to a neuropeptide called arginine vasopressin. And this binds to a receptor called vasopressin receptor 1A. So what about humans, right? We're closely related to meadow voles that we also have arginine vasorepressin, and we also have the vasorepressin receptors 1A. So from what we know in humans, the actual sequence of the receptor, if we look at the genome, the genomic region that contains this gene, does not differ among humans. However, there's a flanking regions upstream of the receptor that shows variation among humans. And this region is likely involved in the gene expression of the receptor and may therefore impact the phenotype that is expressed by the receptor, right? Because we might be expressing more or less of this particular receptor, and this may affect human behavior. So just as a reminder, right, vasopressin 1A, vasopressin receptor 1A, is an actual gene. And genes are actual concrete physical entities that have a location in time and space. So right, if you look at eukaryotic organisms, right, they have the eukaryotic cell contains a nucleogenome at the nucleus, and within the nucleus we have chromosomes. And of course, if you have all eukaryotes also have mitochondria. They have a circular mitochondrial genome, and plants and other photosynthetic eukaryotes also addition have a chloroplast that also contains a excuse me, a circular genome and also contains additional genes. But on these chromosomes, right, the genes do occupy actual spots and places on the DNA molecule. And humans and many other organisms are diploid, right? So that means there are two homologous chromosomes, and therefore we have two copies of the same gene. And those two copies can either represent the same or a different allele, and that's just a different form of the same gene. So alleles are differentiated by differences in the sequence of the genes. So for our study here, the authors whose study I'm showing you used different numbers to show the variation in the flanking region. And so there was one allele that they called the 334 allele, and then they compared that that seemed to have an effect, and then they compared that to sort of any other allele that is around for this particular locus. So if we basically clump all other alleles into another allele and have the 334 allele, we have three different possible genotypes, right? So an organism could be homozygous for the 334 allele, could be heterozygous, or it could be homozygous for another allele. And the other thing is that all cells, right, carry almost, we'll learn about important exceptions, the same genome. The differences just come about by how these different genes are expressed. So if we wanted to genotype a person for the genotype at the vasopressin receptor 1a, we could take skin cells, we could take blood cells, bone marrow, nerve cells, hairs, um, whatever. We should basically be expecting to find the same genotype in all these tissues. So there was a study that was done in Sweden. There was a big data set of medical study in which they had all kinds of so phenotypic information, health data, survey data, and they could link that up with genomic information on the participants in the study. And so in this particular case, they were interested in the effects of the vasopressin receptor 1 on relationships in humans. And so this is just one part of the study that I'm picking out here. But one question the participants were asked, these were all married men and women, have you experienced marital crisis or threat of divorce during the last year? And well, the answer could either be no or yes. And then it was compared to the number of 334 alleles that these individuals carried. And so this is a good example of explaining the concept of genotype, which is the genetic makeup of the organism. So either we have a homozygote for a different allele, a heterozygote for a different allele and the 334 allele, or a homozygote for the 334 allele. And then we have the phenotypes, right? The specific forms of a trait. And we define a trait as the specific character or property of an organism. So in our case, it could either be, and the answer could be no to the question of marital problems, or it could be yes. So just animated explaining what I just told you guys. So, and these are the results from the study. And take a little bit of time to read these results. So we have the raw number of participants that answered the questions for each of the genotypes as these numbers here. So for instance, most participants in the study actually did not have the 334 allele. It was only around about 320 that were heterozygous, and round about 40 or so that were actually homozygous for the 334 allele. And so to compare the numbers a little bit better, I've calculated the percentages in brackets. And what you can see is that the whether you're homozygous for a different allele or heterozygous does not change your answer, right? In both cases, it's about 15% that answered yes, they had marital problems. However, in the case of individuals that are homozygous for the 334 alleles, and these are all men, that proportion doubles. Now it is more than 30% of the people who are questioned. And the researchers did some statistics on this, so this difference is actually statistically significant. So it appears that the 334 allele does have some effect on sort of pair bonding behavior in humans, but it of course comes with a lot of questions, and those are questions I'd like to discuss on Monday during our online class. The other effect that I'd like to the other concept I'd like to introduce is the concept of absolute versus relative risk. And this is sort of a very central concept to any sort of genetic testing that you may encounter, and you will encounter it in your future as these techniques become more and more prevalent. So what you have here in red are the absolute risks for marital problems. So this is basically for the 3 different genotypes, these are the probabilities as estimated from this study of developing marital problems if you carry a certain, if you have a certain genotype at that vasopressin receptor 1A. So again, 15%, 16%, and 34%. Another measure is relative risk. And relative risk is basically expresses the different the risks of different populations that were exposed to different factors as a fraction of some sort of baseline risk. So and often it's the relative risk is calculated in this form. It is the probability of the event in the exposed population in the numerator, and the probability of the event in the unexposed population in the denominator. So in our case, it would be 34%, and exposure here would mean being homozygous for the 334 allele. And unexposed would mean being either heterozygous or homozygous, not a 334 allele. What we're seeing here is that, in the case, it is basically 34% by 15%. We end up with a relative risk of about 2.2% or 2.3%. So in words, what that means is that if you're homozygous for the 334 allele, you have a 230% higher chance of you have a 2.3 times higher chance, let's put it this way, of developing marital problems than if you carry some other combination of alleles. And again, we will discuss how we can basically interpret these results and whether we should take them at face value. But it is really important to distinguish between absolute and relative risks. So because relative risks can be really high, right? Even if the absolute risk is very low. So if you had there was a 1 in a million chance of developing some sort of condition if you underwent some sort of behavior, and there was a 1 in a billion chance in the general population. Well, that would mean the relative risk is actually 1,000 times higher. But the absolute risks for both of these are really, really low. And so you shouldn't really be worried about this. So in this case, actually, the relative risks the absolute risks are actually pretty substantial. So going from 15% to 30%, right? That is quite an increase. So what I hope to touch upon in the discussion of this example is actually the relative impacts of genetics versus the environment. And what I'd like to do in this last part of the lecture is basically look at the relationship between genetics and other sources of phenotypic variation. And so in order to explain this, I'm going to use this, this model here. And I basically took the data from the table, and on the left here, these boxes here represent the genotypes, so 0, 1, or 2 number of 334 alleles. The boxes on the right represent the phenotypes, so either no or yes. And the thickness of the arrows indicates essentially the absolute risk of one of these outcomes, right? So the absolute risk for no is always higher, right? The probability of saying no is always higher for all of these 3 genotypes. It is just that for the homozygotes for 334, they have twice, so the arrow is twice as thick as these arrows here, twice the chance of experiencing marital problems than the other 2 genotypes. And what we're seeing here is an example of a complex trait, because what is going on here is that the genotype does not automatically determine the phenotype, right? If that were the case, it would actually, and I'm going to come back to this, to the previous slide, it would actually look like this, right? So if genotype were a perfect predictor for phenotype, then all individuals that were homozygous for 334 would say yes, and all other individuals with other genotypes would say no. And as you may know, right, this is highly unlikely that a phenotype like marital problems would be so clear-cut, would have such a clear-cut prediction by just genetics without any sort of environmental impacts. So let's go back to this slide. So we can also express the same idea in the following, in which we have, and this is a genetic model in which the genetics determines the phenotype without any environmental influences. That means we have a genetic blueprint either of plan A or B, and regardless of our environmental input, like here different environments 1, 2, 3, plan A, if the organism carries genetic plan A, it'll turn into phenotype A, and if it carries genetic plan B, it'll turn into phenotype B. And this is actually the case for many Mendelian traits or diseases. So for instance, cystic fibrosis, Huntington's disease. However, things are never as simple as they seem. And even in cases like this, there can be environmental factors and interactions with other genes that cause more complex outcomes. So the question is, why is there only imperfect correspondence between the 334 genotype and the phenotype? Well, as you may have guessed, like environment may play a role, but also other genes, right? So because genes are not alone in the genomes, there are more than one gene in the genome, and the human genome has around about 22,000 genes. And so what happens, for instance, if there's additional genotypes here? So in green here, these are the genotypes add a second gene. And in yellow, we still have the genotypes at the vasopressin receptor 1A gene. And under this simple model, we could simply say, okay, the only time you say yes is actually if you are homozygous for the 334 allele, but you're also homozygous for an allele at a second locus. So there's got to be some sort of compounding effect of these two alleles. And since we only tested at this locus, we're still seeing some variation. But it may be that the saying yes is actually completely dependent on the genetics. It just turns out that our genetic predictor was imperfect, and we needed to take into account a second genetic locus. And you can see that, even with two loci, right, this already becomes pretty complex. If we added one or two more loci, right, we'd get a lot more combinations, and this would actually allow for a lot more complexity, even without environmental influences in this case. But of course, right, organisms don't just live in a void, and it's not only genetics that drives phenotypes, but environmental influences also play an important role. So this is our current environment, or wherever you are at this point. So Bellingham, Washington, or the Pacific Northwest, or somewhere else. And under our additional simple model, what's called model 2, only the environment actually explains variation in phenotype. So here we would have environment A that has all these environmental factors. And if an organism grows up in environment A, it'll turn out to have phenotype A. If an organism grows up in environment B, it will turn out to have phenotype B. Genetics is still there, but the genetics does not contribute to the variation we see among organisms. The genetics is just there to basically set up the developmental rules, how the environmental phenotypes, how the environmental influences are turned into phenotypes. here's a really cool example for this. This is a little caterpillar called Nemorea arizonaria, and it feeds on oak trees. And depending on where the little caterpillar feeds, either whether it feeds on leaves or it feeds on these oak flowers, these oak catkins, it'll develop a different phenotype. What you're seeing here in picture A and B are actually full siblings of the caterpillar. So this caterpillar here looks like a little rib of a leaf, right? So pretty well camouflaged, but we can still see it. But somewhere in here is actually its full sibling that has developed the camouflage that makes it look like an oak catkin. And right, we can't clone these caterpillars, so having full siblings is the best thing we can get. But still, um, it is really highly unlikely that this would be caused by any sort of genetic differences between these organisms, especially since it's been repeated and repeated in multiple families. It's basically just the place and the type of food where these caterpillars were reared determined their phenotype. And it's a mechanism by which these caterpillars ensure that they can adapt, so develop the optimal camouflage for the place that they're growing up in. And so it's the phenotype is exclusively determined by the rearing environment. But then, of course, life is never this easy, right? So most phenotypes probably have something like this going on. And in this case, we have the genotype and the environment interact to cause phenotypic variation. So in this case, it is both the genotype and the environment that plays a role. And most importantly, it is the specific combination between those two that actually plays a role. So for instance, genotype 1 will produce a different phenotype, phenotype 1 and phenotype 3, depending on whether it's reared in environment A or environment B. But it is still different also from genotype 2, even though genotype 2 was also reared in environment A, because genotype 2 in environment A turns into phenotype 2, or phenotype 4 when it is reared in environment B. And unfortunately or fortunately, this is basically probably how most traits work. And certainly many traits that we know of have this relationship. So we really cannot see the genotype in isolation of the environment and vice versa. So just to put that back into our example, right? So for instance, here in this case, we may have a very simple case that, well, we have our 3 genotypes, and yes, genetics plays a role. It's only genotype 2 that may run into marital problems, but it only does so if it is exposed to environment B, which could be like a stressful time in their life, right? Like being stuck at home due to coronavirus social isolation. And of course, right, we can always stick both factors together. We could have multiple genes and multiple environments, and you see that the number of possibilities becomes really rather endless in this case. It's sort of a typical property of biology that you take some very simple principles, and by just compounding and adding them and multiplying them, you end up with some really complex patterns. And as I said, most traits are such complex traits, which makes it rather difficult to determine their the genetic basis of these traits, right? One of the jobs that we have as geneticists. And those things could be are important agricultural traits. It could be traits that are with relevance to conservation biology, for instance, how birds can adjust to flexible and unpredictable climates due to climate change, or it could be something like diabetes, which is an important complex disease in humans. So I'd like to end this lecture with the concept of heritability. And heritability is a measure to help us get an idea of how much genetics versus the environment plays a role in the development of phenotypes. And one of the easiest ways to determine heritability, and you will learn more about this in evolution class to get more details on heritability. One of the easiest ways is to work with cloned individuals. And that basically means that we're making replicates of the exact same genotype, right? So we could, in plants, cloning is often very easy because we can simply take cuttings from the same plant and propagate them as separate individuals, and then take these individuals and expose them to different environmental treatments. And in humans, right, cloning humans is very unethical and also probably rather difficult. But there are naturally occurring clones, and we call those identical twins. And in fact, studies with identical twins have been one of the major sources for estimates of heritability in humans. But the principle is that the phenotypic variation that we observe between genetically identical clones must be the effect of environmental variation, right? Because if two individuals are the same genetically, the only possibility, the only other factor that can cause variation in the phenotype is really the environment. And so this is a case that we would describe as high heritability. And what you have here on the x-axis is the phenotypic value. So we could say, for instance, like plant height or body height in humans or some other sort of organism is our phenotype. And then each row is a different genotype, right? Genotype 1 through 6. These are just different genetic variants. And in this case, right, genotype 1 has the lowest mean height with some variation around it. So the little downward stroke here is the mean of this distribution, and this is just the variation of different samples of the same genotype that were grown that basically reflect the influence of the environment. And you see that the means differ between those genotypes, right? So we have one genotype 1 has on average, like a low height, and then genotype 2 in green here has on average the highest height, and the variation is actually pretty close to the mean. So even the tallest individual of genotype 1 is a lot less tall than the smallest individual of genotype 2 here. And so this would be a case in which the heritability is high. That means the environmental variation, right, the variation around this mean here, is small relative to the genetic variation, which is not the variation among the dots of the same color, but the variation that we see among the means of these individual distributions. So again, animated, this is the genotypic variation, and this is the environmental variation for one of them. So here is a case of low heritability. And you can see that the variation between means is very low, right? They're basically all the same. And most of the variation that we see among individuals here is actually due to the environment. Because again, genotypic variation now is a very small range, whereas the environmental variation has a very large range. And so we can actually express this as a formula, and we call this heritability. And we can define heritability as the proportion of the phenotypic variability in a population that is attributable to genetic variability. And we can express this mathematically, and it's called h squared. The squared has nothing to do, well, it's sort of more an historical artifact. It's you're actually not supposed to take the square root of this or to solve this. This is just the way the heritability has been denoted historically. So h squared is the genetic variability divided by the total variability. And the total variability is the sum of environmental plus genetic variability. So let's take a look at some estimates of heritabilities, and in this case for psychiatric disorders. And you see, if you look at the numbers, like a lot of these have pretty high heritabilities, meaning that the genetic variation, that the differences among individuals in a population are mostly explained by genetic variation. And this is also an important thing, right? This formula works with variation, with variability. And variation is by necessity a measure of a population. We can't take a single individual and dissect it and say, okay, this individual's like, this high jumper can clear like 2 meters, and 50 centimeters of that jump is actually due to his or her training, and the other 150 centimeters are due to the genetics of that athlete. That's not how it works. The way it works is that the differences among individuals, right? How much of the differences among individuals in the population is basically attributable to genetic variant differences between individuals versus differences in the environment that these individuals experienced. And so if we look at different disorders, we can see that, for instance, schizophrenia has a heritability of 80%. And we would basically say that 80% of the differences among individuals in the population, basically of whether they develop schizophrenia or not, is due to genetic differences between individuals. But there's still appears to be a small portion also of environmental impact. But it basically means that there's a high genetic component to schizophrenia. And the same is true for bipolar disorder, autism spectrum disorder, and attention deficit hyperactivity disorder. Major depressive disorder has about only half the heritability of schizophrenia. And we would basically say that the differences among individuals, so whether they develop major depressive disorder or not, but there is a genetic component to those differences. The environment also plays a major role in this case. So, and then finally, I'd also invite you to watch this video. And that is a video on epigenetics, and it basically delves into the mechanism of how the environment interacts with an organism's genes. And then finally, I'd just like to provide you with a brief course outline. So we will start out after having given you sort of the big 10,000 feet view of genetics versus the environment. We're going to delve into at the smallest organizational levels. We're going to start with DNA, right? The molecule that carries the genetic information, talk about molecular techniques such as sequencing, and we're going to look at genome structure. And then we're going to look at the mechanisms of how genetic variation comes about, mutations, and we're going to also take that as an opportunity to look at cancer genomics, because cancer is caused by mutations, non-inherited mutations in our body. And then we're going to switch to classical genetics. And so basically Mendelian genetics, we're going to start building up from 1 gene to 2 genes with no linkage. We're going to talk about multiple genes contributing to the same phenotype, polygenes. We're going to look at gene interactions, so more complex ways of how genes work together to make phenotypes. And then we look at physical linkage between genes, so genes that are on the same chromosome close to one another. Talk about how we can use that to mapping. and basically going to be looking at a technique called exome sequencing in our second paper. And these, you may have seen that those papers always have a sort of a genomic component to them, and they're really my way of trying to induce some genomics into this class. And we basically then going to return to this beginning lecture by looking at complex traits and the genetic mechanisms that cause them, because after now we will actually have know all the sort of mechanistic details to understand what makes complex traits, and close out on epigenetics. All right, I'm looking forward to seeing you guys in class, and we'll talk to you later.